We're about to start. Dr. Elise Ahn is here, so we can start. I'm teasing. But often the best place to be is around the food. Don't you notice that when you're at home? When the party is going on, everybody sticks in the kitchen. So we're going to start. And I'm going to welcome everybody to Toronto and Vision Quest 2018. Thank you to everyone for being here. My name is Sharon Colle. I'm the CEO of the Foundation Fighting Blindness. And we have people attending from more than 30 cities and towns in Ontario, Quebec, and Canada. So I'd like to welcome everybody here. If you're from outside of Toronto, let's see who you are. All these people. Okay, now the Toronto people, you got to be nice to the out-of-town people. But it just shows the courage that everybody has, that they are attending this great meeting today. And are there many people here who it's their first time at Vision Quest? Okay, they're putting up their hands. About a third of the room is new. And I want to tell you, first of all, just to relax into it, to look around, to say hello to the person next to you, because you're going to make some wonderful friendships today. And I also want to tell you a little bit more about Vision Quest. The Vision Quest educational series is supported by donors, families, and our corporate partners. It's our annual educational meeting. And in fact, we have them in different cities across the country, but we always try and do them in Toronto every year. It's an opportunity for you to learn about the newest educational and research discoveries that are going on, and we have the speakers here today that will be able to enlighten you about those emerging treatments for retinal diseases. So Vision Quest plays a key role in bringing everyone together, and there's a unique synergy that happens with Vision Quest because we bring together patients, families, industry partners, physicians, and scientists. And there's no other opportunity that brings everybody into the same room to be together and to find solutions. The FFB is Canada's largest health charity funding vision research. In fact, we've funded more than $33 million in sight-saving research. And we've also contributed, yes, it's worth the applause. And we've also encouraged and partnered for another additional $10 million in research. So we're serious. But we're serious about our commitment to research because we're serious about our commitment to the people in Canada who support us. And I want to thank everybody. I also want to welcome the gifted ophthalmologists that are with us today. And I want to thank Dr. Joanne Robitaille for coming to be with us from Halifax. And Dr. Ian MacDonald from Edmonton, who will be speaking next. And these people care deeply about you, and that's why they're here. We are also very thankful to our Vision Quest sponsors this year. And I'm delighted to acknowledge our national visionary sponsors. That's Bayer Healthcare, Novartis, Allergan, and AGTC. These are key partners in our fight against blindness. Thank you. We also have a national event sponsor, Mira GTX, our national breakfast sponsor, Shire, and our local sponsors, Spark Therapeutics and Glaukos. Thank you to all of them. We are very very proud of the first-class events that we put on, and it certainly wouldn't be possible without our sponsors because the rates are very much subsidized so that people can get to this meeting. I'd also like to thank AMI, our accessibility partner. Now, AMI is going to be filming throughout the day, and they're creating a special program called AMI Inside, which is going to showcase all the exciting research that's being talked about today. So isn't this great? You're going to be able to listen to some of these what I call the rock stars of research. They're going to be presenting to you, but you have a front row seat in listening to what's happening, and then AMI is going to be filming it for a program later on so that you can watch it later. If you don't have the AMI app on your phone, it's great. And if you don't know how to put it on your phone, just go to their booth, they're one of the exhibitors, and ask them how to do that. And that way you'll be getting all of these really fascinating topics that are constantly cropping up that you might want to listen to. So I also want to talk a little bit about Dr. Elise Ayan. Dr. Ayan was actually one of the people that created the Vision Quest educational series. And we started it in 1998, if you can imagine. That's quite a few years ago. And Elise has chaired many of those meetings since then and spoken at them. And she continues to see their great value to this day. Because Elise has always, Dr. Ayan, I should be more formal, Dr. Ayan has always believed in the power of patience, that knowledge is important, and that in order to engage people in the development of their treatments, you have to keep them informed. The other thing that Dr. Ayan did was she conceptualized and was the first one to think of the power of the patient registry. And she knew that was an important tool to engage in patients in clinical trials and also to give them to be part of their own treatment. So she championed it, she helped fundraise for it, and she continues to be the chief investigator on it and the principal investigator. Thank you very, very much, Elise. And I should say that Vaishnavi is the key person on this. Vaishnavi is going to be on the panel later today and will be able to answer many of your questions. This afternoon, we're going to have two breakout sessions. Dr. Anna Jurasik, who's probably still towards the back of the room, I can see her. She will be presenting on new technologies. You won't want to miss this. And then Dr. Ed Glassman, who is sitting here, is going to be leading a session for families on how they can be supportive. So if I don't know where to go, but if I was a family and more than a few people here, I might split up because they're going to be in separate rooms. They're both going to be terrific. We're excited at the Foundation Fighting Blindness because artificial vision, gene, and stem cell therapies are no longer science fiction. They're no longer science fiction. But we can't do it alone. The task is too large, and that's why we welcome our partners in government and the private sector and our donors. It's too big for us to do it alone, for any of us. Now, as Canada's largest vision health charity, we always ask ourselves, what can we do to accelerate the development and the availability of treatments? What can we do next? These are big challenges ahead of us to deliver these treatments eventually to you, and that's why we're here. But we're undaunted. And I want to tell you just about one thing that we achieved that was so important to things like the patient registry and clinical trials. We challenged the laws in Canada. For 10 years, we worked on the issue of genetic non-discrimination. And The law of the land in Canada is now, was passed last year. It is the Genetic Non-Discrimination Act in Canada. It's against the criminal code for anybody to discriminate against you on the basis of your genetics. Not employers, not insurers, not anyone. And I want to acknowledge the work that that one person did on this, and that's Lauren Marin, who's here today. Lauren, can you put up your hand? There's Lauren over there. Lauren spoke to the government on behalf of the foundation, and I also want to thank Aaron George, who did the same thing. But, you know, you always have to be vigilant. We won that battle, but we also have volunteer constitutional experts who are willing to take that all the way to the Supreme Court if necessary to protect your rights. Right now, it's the law of Canada, and we'll make sure it stays that way. So thank you to everyone who was involved. It was a great fight. Dr. Mary Sunderland is our Director of Research and Education. She'll be moderating today's session. And we're also joined by Anne Morrison, who's our Director of Philanthropy. And Anne has her crew of family with her. And I also want to give a particular thanks to our talented staff who are helping out today. They're here because they want to create the best possible day for you. But I want to acknowledge the incredible hard work of Janelle Steele, who did an outstanding job on this undertaking. This is a big job. We also have an army of volunteers. Don't they look fantastic? I'm hoping that they'll all gradually come into the room and enjoy the sessions as they get people, latecomers registered and people into this, this beautiful facility. But again, more than 40 volunteers are here today, just because they care deeply about you having a great experience. So who else is playing a great role and a critically important role in fighting blindness? It's you. By you being here today, you are making a difference. You inspire all of the rest of us to keep up our hard work. You are participants in our journey, and we couldn't do it without you. So thank you, and give yourself a big round of applause. Now I'm going to ask one of our leadership volunteers to come up to the podium because I'd like to present them with an award, and that's Lorna Rosenstein. Lorna? Yeah, there's steps here, Lauren. There's one step. 2 steps. Another step. Okay, come on over and hold my arm. Hi. Okay, hi.
I can't see you, but hello.
Oh, Lorna can't see any of us, but that doesn't matter. We can see her, and I can read this award, which is the Volunteer Service Award from the province of Ontario. And it's to Lorna Rosenstein. Stein. Doesn't matter. What? Lorna Rosenstein or Rosenstein? Steen. I should know. At the Foundation Fighting Blindness for her 20 years of volunteer service to this organization. Now, Lorna has served in almost every capacity at the organization, from being a member of the board of directors, the national board of directors, to being a speaker and being a volunteer, she still volunteers at the Cycle for Sight, and is a virtual rider, raising countless thousands of dollars for Cycle for Sight and vision research. She makes phone calls and thank you calls to donors, and she is a mentor to many people, a great friend of the organization, our dear friend Lorna Rosenstein. Here's just this. I'm going to give this to Betty. Okay. Here, they're going to turn around.
I do want to say one thing.
Okay, wait, just a sec. Let me just take
I
a picture.
promised myself I wouldn't. But
Yeah. She promised me she wouldn't. Okay.
Well, I just want to say, Sharon can go on about what I've done for the organization. What she didn't mention is what the organization has done for me. Okay? I have RP. I've watched my vision deteriorate. And being so involved with FFB has given me the strength and the hope that even at my old age, there is still hope for me. And there's hope for certainly the next generation. So thank you. I appreciate this.
And there's also a pin here from the government from the Ontario government that Lorna can wear with pride. And now, I love Helen Keller, and I just read a quote this morning which says, alone we can do so little, together we can do so much. Isn't that the truth? And with that, I'd like to introduce our first speaker, our first keynote speaker, and that's Dr. Ian MacDonald, who is working every day to fight blindness, and he has done so for his entire career. Dr. MacDonald is an internationally recognized expert on inherited retinal diseases, and we're thrilled that he has come all the way from Edmonton to be with us today. The FFB, the Foundation Fighting Blindness, is very proud that we have funded Ian's research for many years. But this includes Canada's first ocular gene therapy. Okay, I'm going to say it again, Canada's first ocular gene therapy. And Ian was responsible for that. He surmounted many hurdles, and he's going to talk to us today about that. Thank you. Ian.
Thank you very much, Sharon. Can you hear me? I tend to be rather soft-spoken, so I like the microphone. And as you know, I'm from Alberta, and you know there's that song that goes, I think I'll go out to Alberta. It's good there in the fall. Well, it isn't. It snowed. You've got lovely weather here. You're just amazing. Why doesn't everybody live in Toronto? Well, thank you so much for the opportunity to speak. I hope that you come away from this energized and hopeful. Congratulations, Lorna. I love the hat, too. The RP logo across the hat. I think that was the first logo for this organization, and it's now changed. And Sharon was very right in saying that the FFB has been behind me for many, many years. The RPI Research Foundation of Canada, which was the progenitor of the Foundation Fighting Blindness, gave me my first grant in 1986 of $18,000, and it started my career. So it was very much seminal. And so thank you all. Thank you for your support. Thank you for making this gene therapy trial possible. It was Sharon's initiative that said, you know, I think we should partner with the Canadian Institutes for Health Research and see if we can get a clinical trial going. And so the FFB with the Choroideremia Research Foundation of Canada and CIHR had a call for an emerging team grant, and our team in Alberta was formed to create a gene therapy trial for a specific type of blindness called choroideremia. It's an X-linked disorder, and I believe there are a few people in the audience today. Gene therapy is a new concept. It's not a concept that I wouldn't have thought possible until the last 10 years. I started out my career in McGill, and I was in undergrad in genetics, and gene therapy was something like a moonshot, but it's with us today. And there are many centers doing gene therapy in North America and across the world. And there are a number of gene therapies that are being trialed. There's one for achromatopsia. This is a disorder of photosensitivity. There's one for choroideremia that I'll talk about. There's a gene therapy trial for an infantile form of retinitis pigmentosa called Leber's congenital amaurosis. And that was the trial that enabled the world to know that gene therapy could be done in the eye. It was safe and effective. And it is now a therapy that has undergone approval from the Foundation Fighting Blindness. It certainly has. The Food and Drug Administration of the United States, and the product is called Luxturna. It is a very costly drug, but I'm hoping that the price will come down. There's also a gene therapy trial for Leber's hereditary optic neuropathy, a disorder usually of men affecting the optic nerve. There's a gene therapy trial for X-linked RP, and we have the folks from AGTC here who are actively involved in this trial. There's a gene therapy trial for retinoschisis, for Stargardt disease and Usher syndrome, the type 1B. It's a very complex field with so many names, so many acronyms, but my goodness, gene therapy has arrived, and it has arrived in Canada, and many centers in Canada are actively concerned about linking you as patients to these trials. And we'll have a discussion about the importance of the Foundation Fighting Blindness Registry of patients with genetic eye diseases. Well, how do you create a clinical trial for gene therapy? It took a team to put this together. And this team includes people who have expertise in genotyping, in other words, finding the mutation in the particular gene that is of interest in the trial. People who can build a vector, something that will replace the abnormal copy of the gene with a normal copy. We have people who can do testing with imaging of the retina, measuring outcome measures like the visual field, visual acuity, electrophysiology, and we have genetic counselors who can talk to the patients and describe the process of gene therapy to them. Our translational research team also included 2 surgeons. Both of them happen to be residents of mine. I'm very proud of these 2. Matt Tennant, a very positive fellow, a can-do type of individual, and his colleague Riz Somani. We had 2 surgeons in case one fell ill, and we had to back that up. In order to do the gene therapy, it is a surgical procedure requiring the expertise of confident surgeons who can put a very small amount of fluid containing the vector, which is essentially a copy of the gene packaged in a protein, and developing that technique to put it underneath the retina safely. We've had the support of the Foundation Fighting Blindness, the Choroideremia Research Foundation of Canada, the Canadian Institutes for Health Research, the Canadian Foundation for Innovation, in order to provide the equipment in order to do the trial. Some of the outcome measures that we used required machines that we didn't have, and so we had to buy those machines. And we had to ask our regulators, like Health Canada, to ask for permission to bring this vector into Canada. We had to go to the Children's Nationwide Hospital in Columbus, Ohio, to actually look at the manufacture of the vector to make sure that it was safe, and then get an import license to bring it into Canada. And then there's one thing that we forgot. What do you think we forgot? Well, I didn't know. So it's those unknowns that you didn't find out until after the fact. So there's another level now called Environment Canada. So if you bring something into the environment, like a gene encapsulated in a virus, Environment Canada wants to know because it's going to go into the environment. So we need permission now from Environment Canada as well. We've had collaborators from Alberta Innovates Health Solutions, which is the next generation of Alberta Heritage Foundation for Medical Research, who have funded us as well. So there's lots of money that had to go into this to provide funding for the salaries of the people who are going to undertake the trial. The hospital provided the operating rooms at no cost. The surgeons did the surgery at no cost. I'm free too. So, just kidding. So the gene therapy that we did was for choroideremia. This is a rare disorder affecting mostly men, happening at about 1 in 50,000 individuals in the general population, and it results from the absence of a protein because the gene is abnormal. The gene makes the protein. If we can put a normal copy of the gene in the eye to make the protein, then that protein can function in the normal physiology of the photoreceptors and the pigment layer that is degenerating in the eye. So the purpose of our experiment was to see whether this gene therapy was safe, and it was to measure any difference in between the treated eye and the untreated eye. So we were looking for measures that would say, can we show whether there's a difference? And so for our initial inclusion of individuals within the trial, we wanted to make sure that they had about the same quality of vision in either eye. If they had one eye that was quite different than the other, well, then we wouldn't want them in the trial for the fear of having one eye treated and losing vision in that eye. We also wanted to have no other problems with the eye that we weren't treating, so that there was no gross asymmetry in between the two eyes. We treated the worst eye with the worst vision, though, just because I think the patient would want that. They wouldn't want their best eye to be treated. We looked at 20 candidates. These were from the Foundation Fighting Blindness Registry of patients who had registered for future clinical trials, and also our own database of individuals. And we showed those 20 to 4 vitreoretinal surgeons and said, which ones do you think you would choose? So we tried to unbias ourselves. I couldn't handpick the people that I thought would be eligible for the trial. And it was amazing that these 4 vitreoretinal surgeons, 3 from Canada and Robert McLaren from Oxford, a bit of disclosure there, he was our colleague in Oxford who had started the first clinical trial for choroideremia in Oxford in England. And they all agreed on 6 individuals. And I know that some of you are in the audience that have good vision, and even for you with good vision, this is a big test, because this is a snapshot of what the people signed up for in the clinical trial. They had to undergo preclinical testing. They had to be genotyped. They had to have specific measures of their vision tested and repeated so that we knew that they could undergo the tests. And then they had to have the surgery and be tested right after the surgery, 1 day afterwards, 1 week afterwards, 1 month afterwards, 3 months afterwards, 6 months afterwards, 9 months afterwards, a year afterwards, and so on, with a number of tests. So the patients really had to be committed to this. It was a big undertaking for us as a small team and for the patients. They were all Albertans. They were different sizes. They were all men, different races, and by chance, there were 2 brothers. We learned a lot by doing the clinical trial. We learned that visual acuity wasn't necessarily the outcome measure that was going to tell us whether this was an effective trial. Here's an example where the untreated eye got better during the course of the therapy. Darn, how did that happen? But it's simply because the patient began to be well trained in how to take vision. And so it wouldn't necessarily be an outcome measure that you would think that the untreated eye would get better, but his did. We also tested the area of the pigment epithelium that would remain in the patient after the treatment in the treated eye and the untreated eye. So unfortunately, this is like a rug in the back of the eye of single cells. They're pigmented cells. These are the cells that support the photoreceptors, and they unfortunately undergo degeneration, and there's a margin that gets smaller with time. And this was a very, very valuable measure. And it's going to be a valuable measure for future clinical trials for gene therapy. In this particular case, the area of retinal pigment epithelium in the treated eye and the untreated eye followed the same course over a period of 2 years. We've published this material in the past month in the American Journal of Ophthalmology, and it dates from the end of the trial in 2017. So this is a 2-year trial, and the results of 2 years are available, and now we have 3 years of data. And it seems that the treated eye and the untreated eye, in terms of the area of remaining pigmented layer, shrank at the same rate. That was not an expectation of ours. And unfortunately, one individual experienced a significant inflammatory reaction within the retina, something that hadn't been predicted in some of the preclinical work that had been done, and also the first trial by Robert McLaren. So this was a great shock to me. And he was treated with high-dose steroid to prevent the inflammation, and had to be repeated with another dosing of high-dose steroid, and despite that, lost vision in the eye. He has his other eye, which is okay, but one would not have expected that this would have happened. So it's a cautionary tale for all of us to know that these treatments that require a subretinal injection can have two effects. It can be damaging because of the detachment of the retina, and it can also trigger inflammation for which there may be a permanent loss of vision. So really, those were the takeaway points that I wanted you to know as an organization, as patients, as family members, to know that these are still experiments. But nevertheless, we gained very important information for colleagues across Canada and also across the world to know that this was the outcome of the trial. Maybe we can do things a little bit differently the next time, because we keep saying to ourselves, we've got to move on from this and get better the next time. So are we looking for things to do differently? Yes, most definitely. We're looking for alternate vectors, maybe ones that don't have what we call a backbone of a virus to get it into the eye. And we're obviously interested in expanding these trials to include other diagnoses. So we learned that we can participate in ocular gene therapy here in Canada, and I'm sure there are other centers. I know there are other centers than Edmonton, definitely here in Toronto, in Halifax, in Vancouver, and elsewhere, who are actively wanting to partner with industry in enabling patients to access clinical trials of gene therapy. It takes a team to do this, though. This is not all about me. It's all about the patient and their families, but it does take a team, and it's a team of committed individuals. If somebody gets sick, you have to replace that individual. If somebody moves, you have to replace that individual. You've got to put that team together so it's going to stay together for a period of the duration of the trial and longer. And just as Lorna was talking about hope, it's given me great hope that we can do this here in Canada and that we can bring clinical trials, meaningful trials, to our patients. And so I wanted to tell you just briefly, because I'm sure I'm running out of time, that at this point, this is still a research endeavor. We have still hope, but these are not treatments at the present time because we don't know if they're truly safe or effective. It's still an experiment, and there are risks that you need to take into account, and we should be clearly communicating those risks and having a clear understanding, I think, on the other side. And the timelines are long. We started our clinical trial 3 years ago. We took a year to write up the results, and we're still looking at the patients for the future. For those of you who can see, here's a little snapshot of those grinning faces at the end, and it was nice to take a snapshot. For those of you who have really good vision, there's only one nurse in the middle, and some of the surgical team is marked there, Matt Tennant, Riz Samani, and then there's Helen Highwaters. Too subtle a joke, I guess. Okay, so got it, okay. So it's not Helen Highwaters, but we needed a very good surgical nurse to keep us on track. So thank you very much, and I'll step away from the microphone, but I won't drop it. I think at this point, we're going to have a panel discussion, and I would invite Elise Hayon and Joanne Robitaille and Vaishnavi to come up. This is the first time that I've met Vaishnavi, so for us, it is, just as for you, a very important time to meet people who we've only spoken to on the telephone.